Welcome to CliPP-On-Web, a browser-based interface for the CliP algorithm, clonal structure identification through pairwise penalization. This tool lets you reconstruct the subclonal architecture of tumor samples from bulk sequencing data without needing to install any software locally. Today, I'll walk through each section of the application. When you first open the app, you land on the About page. The description at the top summarizes what CliP does. Below the description is a pipeline overview showing the 4 steps CliP takes. Further down, you'll find the citation for the CliP preprint with one-click buttons to copy the BibTeX entry or download it in RIS or EndNote format. Now, how does CliP work? If we click CliP-On-Web in the sidebar, this is where we can run CliP on our own samples. CliP requires 3 files per sample: an SNV file, a CNA file, and a purity file. Example files for each are linked at the top of the sidebar panel, so you can see the expected format before uploading. You can either upload all files at once or upload them individually, sample by sample. Set the number of samples you want to analyze. Then we set the samples. File upload fields will appear here dynamically. Once the files are uploaded, we can click Upload Samples. To run the analysis, we just select the sample we want to analyze from the dropdown, then click Submit. When it finishes, 2 plots appear in the main kernel. The VAF plot, which is a kernel density estimate of variant allele frequencies with individual mutations shown as points on the bottom, colored by clonality, and the CP plot. It's the same view, but for estimated cellular prevalence, with dashed vertical lines marking the identified subclonal clusters. You can adjust the smoothing factor for each plot using the sliders. This adjusts how smooth or granular the density curve appears. You can also switch point coloring from clonality to red depth, and vice versa. Click Clear to reset and start a new analysis. For data resources, we can click CliP Data Resources in the sidebar. This section gives us access to pre-computed CliP results for large public cohorts without needing to run anything yourself. There are 2 subtabs, TCGA and PCAWG. TCGA contains results for 7,279 samples spanning 33 cancer types. We can use this drop-down on the left side to select a cancer type, then a specific sample, and click Submit. The same VAF and CP plots appear in the main panel with smoothing and color controls as well. After the plots load, we can download VAF plot and download CP plot so we can export these files directly. This table in the sidebar shows how many samples are available per cancer type. PCAWG works identically, covering 2,778 whole genome sequence samples from the Pan-Cancer Analysis of Whole Genomes project. Both tabs note that samples pass filtering criteria, more than 20 and fewer than 20,000 SNVs, and an NRPCC value greater than 10. Now on to driver mutation. This page lets you explore driver mutations across 5,440 TCGA samples, covering 289 genes from 31 cancer types. The driver gene list comes from Intogen. Start by selecting a cancer type from the dropdown at the top. A table loads showing all driver mutations for that cancer type, by default an individual mutation view with columns for gene, protein change, consequence, cancer type, clonality, VAF, and CP. Once we have clicked on a row in the table, we can see the samples VAF and CP distribution plots appear below, giving us the full subclonal context for where that mutation falls. Smoothing control appear, for both plots once a row is selected. At the bottom of the page, two buttons become active once you've selected a row. Download by sample, which exports all driver mutations for that sample, and download by gene, which exports all mutations in that gene across all samples in the cohort. Finally, we have the Downloads tab. This page provides direct access to the underlying datasets. There are 4 cards here. First, the simulation dataset, or CliPP-Sim4K, which is 4,050 simulated samples with known ground truth useful for benchmarking. You can download the 2 MB summary TSV or the full 212 MB zip archive of individual sample files. There's TCGA, which you can download the full 372 MB zip archive of per sample clip outputs, or use the download by cancer types to get a focused TSV of raw mutation assignments, chromosome, position, cluster index, and cellular prevalence for any single cancer type. PCAWG is the same structure as TCGA. We have the full 2.8 GB archive or per cancer raw assignments. PCAWG samples are whole genome sequenced, so the files are larger. Finally, we have the driver mutations. You can download the full driver mutation dataset, which is 16,913 mutations across 5,440 samples and 31 cancer types, as a single CSV with 9 columns: sample ID, gene, chromosome, position, cancer type, clonality, VAF, CP, and consequence. Or use the cancer type selector to download just the mutations for one cancer type. That covers the full application. To summarize, CliPP-On-Web lets you run subclonal reconstruction on your own data through the CliPP-On-Web tab. Explore pre-computed results for over 10,000 public tumor samples through the Data Resources tab. Investigate driver mutation clonality patterns through the Driver Mutations tab, and download the underlying datasets directly from the Downloads tab. For questions, use the contact information on the About page. Thank you for watching.